Hello and welcome to another episode of Odds and Ends. In this episode, we'll be talking about rice domestication. For the longest time, there's been an ample lack of genetic evidence on how domestication happened in rice, with most researchers relying on archaeological data to make determinations on how it happened. In 2022, however, Dr. Takeshi Izawa at the University of Tokyo did an extensive genetic analysis of the various cultivars and wild specimens in rice to determine the full nature of domestication within this species. The early challenges done were finding related QTLs within rice. The first known examples of these traits was discovered in 2008 by Dr. Shoma et al., with further traits discovered in 2017 by Dr. Liu. Dr. Izawa did extensive research within the land races of rice and found 6 domestication-related quantitative trait loci. The most primitive cultivars, those belonging to the tropical Japonica subgroup of rice, possess most of the wild traits found in the wild species, suggesting that these old cultivars are closely related to the original rice cultivars in ancient times. Although suggestions that these wild traits may have been integrated from wild rice back into domesticated rice at some point in history. These two hypotheses contradict the current archaeological analysis. Further research studying the variations between 3,000 rice cultivars amongst all land races and modern groups discovered at least 4 major bottleneck selection events happening within rice, which were then put through a smaller bottleneck to create the 8 subgroups. In 2004, Dr. Vaiti et al. Discovered that through the tracing of various retrotransposons that are highly heritable, it was likely that the major diversion between the Japonica and Indica strains of rice occurred 400,000 years ago. This was earlier than expected date, and it contradicts another piece of evidence which suggests that rice was domesticated years ago. These two methods of analyzing genetic data found that it was likely that the Japonica and Indica lineages of rice were both domesticated independently. The question of how the selection process happened, it was found that there was a high level of genetic bottlenecks that happened in rice domestication. Based on its low genetic variability, it's speculated that rice was initially gathered from the wild, and as people dropped seeds here and there, they would grow into rice plants near the homes of people. And it was speculated that this population became the origin point from which rice was domesticated. This may explain the origins and genetic bottleneck of domesticated rice. There's only a small number of haplotypes, which are a small group of genes that are always inherited together within rice. This indicates a very low genetic diversity and heavy-handed genetic fixation, indicating that the original population that rice was domesticated from was quite small. There are several genes that seem to be the origin points of the domestication process. GHD7 controls the flowering period, and mutations within that gene cause its earlier flowering time, making it acceptable for ancient peoples as a food source. The genes HD1 and EHD1 confer prolonged growth until flowering, which further increases yields. Several integration events have been found in rice. The wild rice has black seed, controlled by the COLA4 color gene. Most domesticated rice has white seeds, controlled by the RC gene. Various studies and textual documents showcase domesticated rice, but with black seeds. Within the Japonica and Indica lines, there's various ancient cultivars that also have black seed coloration, a very distinct wild trait. This indicates that that gene was incorporated back into domesticated rice through crossbreeding between both the wild and domesticated lineages. This likely happened at least 1,000 years back. The genetic analysis combined with the archaeological analysis suggests that, over the course of 1,000 years, wild rice and the Indica lineage of domesticated rice led to several traits including black seed color to be integrated within the Indica subgroups. Since there's later evidence of integration of the black seed trait into tropical Japonica lineages, It is likely within those lineages of black-seeded Japonica cultivars were the result of crossbreeding between these indica lines with black seed and tropical Japonica line. These would have been crossbred into modern temperate Japonica cultivars. Cola 4 is a full locus, or large section of a genome that contains the genes for black seeds. Since this trait gets passed down quite easily, it is plausible that it's controlled by one gene. The large dataset obtained by these researchers, in combination with the research done by other groups, have created a very comprehensive model for rice domestication. The Japonica group was the first group to be domesticated, and it went through a very large bottleneck, indicating that only a small number of plants were used in the domestication process. Its population would then divide into 3 different groups: subtropical, tropical, and temperate Japonica cultivars. Around the same time, the indica population would be domesticated, and subsequently mixed with wild rice to breed different varieties. With this data in hand, a single or multiple origin for domesticated rice is possible. Right now, the 2-origin model of domestication is the most likely one. There's great genetic differences between the indica and Japonica lines of domesticated rice, so both lineages were likely domesticated from different populations of wild rice. The genetic evidence also suggests that the indica and Japonica lines have a genetic distance that separates them by 400,000 years, while domestication took place between 8,000 and 9,000 years ago, based on archaeological evidence. This suggests a genetic divergence and possible subspecies formation around 400,000 years ago within wild rice, with different groups domesticating different subspecies or subpopulation, with layer mixing between both lineages, as well as mingling between wild and domesticated lineages. There's still several questions that remain to be unanswered, however. How many intergressions happen? What are the full genetic differences between the wild rice and domesticated rice? There's still a great deal of information that determines some interesting realities. Between 17 and 15% of the genome was intergressed from the Japonica line into the indica line, in much the same way that the black seed trait was intergressed from the indica lineage into the Japonica lineage, indicating later iterations of crossbreeding between both populations. But with the current research and hopefully future genomic studies on wild rice, many of these mysteries will be solved, and a fuller picture of the domestication of rice will be made clear. And that about covers everything. Thank you for watching. Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in the description below. Thank you for watching.